Hello. Thank you all for joining this talk. I'm Claire Zhou, the product manager for our CRISPR and oligo services here at GenScript. And it's my honor today to host this session with Dr. Kong and discuss the development of a T-cell engineering platform for fully individualized neoantigen-specific TCR therapy. Dr. Xiangjun Kong is a principal scientist at Neogene Therapeutics who has extensive research experience in high-throughput functional screening and genomic editing technologies. Before joining Neogene, he was a postdoc fellow at the Netherlands Cancer Institute in the laboratory of Professor Daniel Pieper, where he co-developed and published several novel targeted and immunotherapy strategies for cancer. He received his Ph.D. in cell biology in the laboratory of Professor Tao Zhu from the University of Science and Technology of China. And without further ado, I'll pass the floor to Dr. Kong.
Great, yeah. Thanks for the kind introduction. So, my name is Kong. So I'm a principal scientist from Neogene Therapeutics. So it's my great honor to present our development of Neogene Therapeutics for T-cell engineering platform for the fully individualized neoantigen-specific T-cell therapies. So one of the key genetic differences between the tumor cells and the normal healthy cells is the tumor cells that have accumulated the mutations in their DNA sequences. So some of these mutations that can generate new antigens. So these new antigens that can be processed and presented by imaging molecules on the tumor cell surface and be recognized by the T cells which have a specific TCR sequences. So these TCR sequences, they have therapeutic potential. So normally in the TCR therapies, so people will engineer the patient's own T cells to express these therapeutic TCRs and infuse back to cancer patients to fight cancer. So as most of these new antigens, they are patient specific. So which means the TCRs recognize these new antigens is also patient specific. So therefore require a fully individualized therapeutic approach to targeting these new antigens. So at Neogene, so we are developing a fully individualized TCR therapy platform. So in brief, so we obtain the tumor biopsies from hospital, and then we perform exome sequencing to identify the tumor mutations. So at the same time, perform the TCR sequences to identify the TCR sequences within the tumor. So by performing some high-throughput functional screening experiments, so we can select the TCRs which can recognize the new antigens. And then we engineer the patient's own T cells to express these therapeutic TCRs and infuse back to the patient to fight the cancer. So as you can see, so this is a fully individualized therapy, which means the conventional T-cell engineering methods, so such as using the retro lentivirus, is extremely unlikely to apply here because it just takes too much time and costs too much to produce a patient-specific virus at GMP level. So therefore, a long viral DNA-based delivery methods it requires for targeting these new antigens. So before so for people who are familiar with the genetic engineering, so you can already guess we are using genome editing tools, so such as CRISPR Cas9 to introduce the therapeutic T cells into primary human T cells. So the way we engineer the T cells is like this. So we obtain the buffer codes from the vendors, and we isolate the T cells and activate these T cells. So after activation, so we electroporate the T cells with the Cas9 together with the guide targeting GRAC. So this is the gene encoding the TCR alpha constant region. So this RNP complex, once enter cells, so they will behave like a scissors that will create a double-strand break into a TCR alpha constant region. So at the meantime, if we provide a template which contain a therapeutic TCR sequences, and this template can guide the cells to repair, and then the therapeutic TCR sequence will be incorporated and expressed into the T cells. So from this experiment, you can see, so before editing, you can see the most of cells, they are endogenous TCR positive. So 94% they are expressing the TCR constant beta sequences. So if we electroporate with the TRSC RNP complex, you can see the TRSC will be disrupted, therefore the most cell will become TCR negative. At the meantime, if we also include the repair templates, in this case, this repair templates produced by the PCR amplification and encode the V-beta 13 positive TCRs, you can see 24% of the cells were expressing therapeutic TCRs. So we performed a lot of this TCR editing across different experiments and different doses. So on average, you can get around 20% of the editing efficiency. But you can see on the right, the efficiency varies from a low of 5% to a high of 40%. So all this works fantastic, I mean, in the lab. But before we can apply the TCR editing to clinical TCR products, we have several challenges that need to be solved. So the first one is to identify the TCR edited cells. In the previous slides, I show you we can use the V-beta antibody to track the TCR edited cells, but not all. Actually, most of the TCR they cannot be be tracked by the antibodies, because the V-beta antibody just does not exist. So therefore, we have to develop novel approaches to identify the TCR edited cells. And the second is the TCR mispairing. We introduce the therapeutic TCR into the TCR alpha region, so therefore endogenous TCR alpha will be disrupted. However, endogenous TCR beta chain is still expressed and mispair when I introduce the TCR alpha chains. And the third one is to standardize the TCR editing efficiency. As I just showed in the last slides, the TCR editing efficiency varies across different donor and experiments. And this limits the therapeutic, standardize the therapeutic products. And also to maximize therapeutic efficacy, we want to have very high pure of the TCR edited cells in our TCR products. And the fourth one, so right now we're using the DNA repair template produced by PCR amplification. So this is definitely not going to be a GMP applicable. So therefore, we have to find the GMP grade DNA repair templates to perform the clinical manufacturers. So the first one, to identify the TCR edited T cells, so the approaches we are trying to achieve to introduce a unique antibody epitope to the TCR sequences and use the antibody to detect the TCR edited cells. So the current kind of the status to track the TCR edited cells to engineer the human mouse hybrid TCR and use anti-mouse TCR constant beta antibody cloning H57 to track the T cells. So you can see from these FACS experiments, on the left is a fully human TCR sequence, TCR edited cells, which means the variants and both variants and constant they are from human sequences. Therefore, they cannot be detected by H57 on the y-axis. So if we edit T cells to expressing the human murine hybrid TCR, which means the variant is still from human sequence, but the constant region is from fully mouse sequences. You can see now the TCR can be recognized by the H57 antibody. However, this approach has its limitations. Introducing the murine constant sequence will increase the size of transgene, so which may reduce the editing efficiency, and also the murine sequences will increase the risk of the immunogenicity. So the approach we are taking is to determine which amino acid epitopes this antibody recognizes, and then introduce this minimal epitope into the TCR constant domains. So according to the literature, the H57 antibody interface is mainly located on the FG loop. So this is the loop sequence in the TCR beta constant region. So this is the main kind of interface between the antibody and the TCR sequence, and also some of the amino acids at adjacent A and B strands are also play rules into the recognition of the antibody. So we have maps between the human TRBC and the mouse TRBC sequences, and we found there 10 amino acids are different between human sequence and mouse sequences. So therefore, we replace these 10 amino acids from human, replace with a mouse counterparts, and create a variant called mu-ABFG, which is this A and B strand from murine and FG loop from murine, and then editing T cells to express this 10 amino acids from murine and modify the TCR sequences. You can see from these Fox plots, the fully human TCR, they cannot be recognized by this H57 antibody, which is on the y-axis. Once the TCR was modified with mu-ABFG, 10 amino acids from murine, you can see now the TCRs can be recognized and be tracked by the H57 antibodies. But not all these 10 amino acids, they are important for antibody to bind, to find the minimal epitope. So what we have done is to swap back all these 10 amino acids one at a time and to see which one is essential for antibody to bind. From this experiment, you can see there are only 6 amino acids which are important for antibody bind. So once we swap back to the human counterparts, they can't recognized by the antibody anymore, which are shown by this red white plots. So this we call this 6 murine amino acids mu6, and then if we introduce lots 10 amino acids from murine, but only 6 amino acids from murine into the human TCR sequence, you can see now the TCR still can be recognized by H57 antibody. So which means by introducing only 6 amino acids from mouse TCR sequence, we were able to track our near fully human TCR with H57 antibody. So I think I have shown you the data. So we have one strategy. We can introduce this mu6 epitope into our near fully human TCR and enable the TCR to be recognized and tracked by the H57 antibodies. A second challenge we have to avoid the TCR mispairing. As I just explained, the endogenous TCR beta can pair with introduced TCR alpha. So the approach we are preventing the TCR mispairing to include the second RNP complex to target the TCR alpha, TCR beta constant region to disrupt endogenous TCR beta expression. As I just showed previously, normally if we provide editing, we electroporate the cells with the RNP complex target the TRAC, which will disrupt the TCR alpha, endogenous TCR alpha genes. So now we include the second RNP complex to target the TRBC, which encodes the TCR beta constant region. So therefore, both of the endogenous TCR alpha and beta chains were disrupted, and only the introduced TCR chain will be expressed, therefore can avoid the TCR mispairing. From this Fox plot, you can see if we only include the TRAC RNP complex, you can see most of the TCR edited cells will express both introduced TCR and endogenous TCR beta. So they are double positive, which means a lot of TCR mispairing happens in the TCR projects. If we include the TRAC and also the TRBC RNP complex to disrupt both endogenous TCR alpha and beta chains, we can see the majority of the TCR mispairing are prevented. So I can show, I think I have shown you the strategies and to avoid the mispairing by knocking out the TCR beta chain by including the RNP complex to targeting the TRBC sequence. And the third challenge we have is to standardize the TCR editing efficiency. So we can see from previous slides, the editing efficiency varies between donor and experiment. So we want to make it standardized and also as high as possible to maximize the therapeutic efficacy of the TCR products. So the approach we're taking is to develop drug-based selection methods to enrich the TCR edited cells. So during culture. The approach we're taking is to utilize the DHFR, the methotrexate based selection system. So DHFR, the name is a dihydrofolate reductase. So it's an essential enzyme for mainly for DNA synthesis. So there is a drug called methotrexate. This is a clinically approved DHFR inhibitors, which are used for cancer treatment and also for the immunosuppressive for the autoimmune diseases. So for DHFR, there is a mutant version of the DHFR to have two mutations on the DHFR sequence, and this mutant DHFR is resistant to methotrexate. So now if we include this mutant DHFR into our repair templates, so therefore the successful edited cells will express both TCR and mutant DHFR. And then if we treat with methotrexate, the only successful edited cells will express mutant DHFR, therefore be resistant to methotrexate and continue to grow. And the unedited cells, because they don't have the mutant DHFR expressed, so they are sensitive to methotrexate, they will drop out so during the culture. You can see from this Fox plots. So normally, if we use a normal kind of editing approach without methotrexate, without the DHFR, we can get around 27% of editing efficiency. But now, if we incorporate the DHFR into repair templates and select some methotrexate, you can see the editing efficiency increase to 85%. Of course, you can also imagine with 85% of the cells in your TCR product, they are recognize new antigens. So the T cells also shown a much functional in terms of cytokine production. For example, interferon gamma production. If we culture cells with a cell, we culture T cells with antigen presented cells, which are have the right peptides and expressed. And also this TCR editing in selected cells will show much better tumor control capacity. So you can see on the right, the tumor killing capacity compared with conventional unselected cells. So I think I have just show you the strategy. So we have used the mutant DHFR methotrexate selection methods to standardize TCR editing efficiency and also to maximum therapeutic efficacy. So another challenge we're facing for the TCR editing is to use the GMP grade repair templates. So all these experiments, they are performed with the DNA template produced by the PCR amplification. So this is extremely unlikely to be a GMP compliance, and also it's very difficult to verify the sequence. So from the TCR amplified the TCR products. So what we have done is to collaborate with GeneScript to adopt so they are closed-end DNA repair templates. They call the gene-bound double-strand DNA, which can be produced with the GMP grade. So I just explained the current DNA repair templates, the PCR amplified. So they are easy to produce, but they are not suitable for clinical manufacturing. So as the PCR amplification is error-prone and very difficult to verify the sequence. So we collaborate with GeneScript and to adopt their linear closed DNA, gene-bound DNA, and could be potentially to produce in GMP grades and perform some experiments and to see how it goes in for editing the T cells. You can see normally we use 1 microgram per 1 million cells. You can see works quite good. Without the mesothelin selection, we get 27%, and with mesothelin selection, we get the editing efficiency enhanced to the 85%. But we do observe this gene-bound double-strand DNA is quite toxic compared with a PCR amplified templates. So therefore, we decrease the dose of this double-strand DNA to perform the editing. So if we perform some experiment with 0.5 microgram per million cells and also 0.25 microgram per million cells, we do observe the editing efficiency drops if we use less double-strand DNA, but the cell viability is much higher. So in the end, we get much more cells compared with the dose with a high dose. And regarding the dose we use with the mesothelin selection, we can get above 80% of editing efficiency. So now we have all these tools in our disposal. So for example, we use TRAC, IMP to disrupt the TCR alpha chain and also mediate the DNA repair. So we incorporate the Mu6 epitopes in the TCR sequence in able to track the TCR edited cells with H57 antibody. And we incorporate the mutant DHL sequence into TCR repair templates to enable us to select the methotrexate. And we also adopted potentially GMP applied compliance the double-strand DNA produce gene script to perform the genome editing. So at the meantime, we also disrupt the endogenous TCR beta chain by incorporate the TRBC IMP complex. So this all works very well in the lab in a small scale experiments. So can we scale up to manufacture clinical TCR products? So to prepare for NT125, so it's a phase 1 clinical trial on Neogene, the fully individualized TCR therapy to treat advanced solid tumors. So we collaborate with the Netherlands Cancer Institute, Antonie van Leeuwenhoek Hospital, and they have the GMP facility to produce the manufacturing of the TCRs, and we collaborate to perform the 3 very large engineering runs to prepare for the trial. So we obtained the apheresis from the donors, and we perform the TCR isolation and activation, and electroporate with IMP complexes and double-strand DNA templates, and subsequently we select cells with methotrexate and clonal expansion for around two weeks, and harvest the cells for profiling. So in total, we performed 3 large-scale engineering runs. You can see the FACS plots. All 3 engineering runs at day 14. After manufacturing, we can get more than 65% of the T cells in the T cell products. They are TCR edited. And from all 3 engineering runs, we can obtain about 3 billion cells. So after around 2 weeks, so manufacturing, which is very high, the dose required. And by profiling the cell phenotype, by studying some phenotypic markers, for example, CD45RA, CD62L, CD27S, and we saw that most of the cells, they obtain a stem memory cell like phenotype, and then secrete multiple kind of cytokines. So this is one of the key characteristics of the functional T cells. Of course, once we culture these T cells with the target cell with expressing the corresponding antigens, so we can see the T cells, they're killing the target cells and very well. I think in summary, so we have developed and several tools and applies and these several tools into our TCR manufacturing procedures, and we have collaborated with NKI to perform very successful engineering runs and to prepare for our first clinical trials to treat the solid cancers. So with that, also I would thank some people. So so this is truly teamwork, as most work we performed, the hard work performed by Paula, Asta, Sander, Raquel, Relat, and Bas, and for Mariana, Candy, Thomas, Tong, Carson, Gavin, and Jeroen, and for their support and supervision, and for our collaborators as the Netherlands Cancer Institute to perform the 3 larger clinical manufacturers, and for GeneScript for provides the GMP grade gene-bound double-stranded DNA template. So with that, I would like to finish my presentation, and if you have questions, I'm very happy to answer.
Hi, thank you so much, Dr. Kong, for a great talk. I'm really glad to see these wonderful results with up to like 85% knock-in using our Gen 1 double-strand DNA. And also, I'd congratulate you and Neogene on the May approval of your first phase 1 clinical trial for this fully individualized TCR therapy. So I think we did get a couple questions online, and we should have time to answer some of those. So, and dear audience, if you haven't tapped your questions in, please do so, and we'll select some for maybe another 10 minutes. So I guess we'll start with this first question is, in the tumor killing comparison between TCR and TCR-MTX resistant, have you normalized the transduced percentage? And if normalized, why is the MTX resistant drug product show better efficacy?
So that's a very good question. So the data I showed on the slide, so they are unnormalized, which means in the mesotract select cell products, they have high frequency of TCR-edited cells. So we also have performed the normalization experiments. So we use the same percentage of TCR-edited cells to compare the normal editing approach with the mutant DHL methotrexate approach, and there we didn't see the differences regarding the tumor cell killing. So therefore, the results so we shown is mostly because now in our TCL project we have much high frequency of TCLs they're expressing therapeutic TCLs.
Thank you. And I think a follow-up question. So how many days of the MTX selection is necessary, and how to quantify the residual level of MTX in the drug product?
So we usually only perform very short periods of mesotract selection. So in our standard kind of procedure, we perform 3 days mesotract treatments. So after that, we just stop the treatment. Yeah. So to profiling the residual mesotrax in the cells or in the medium, so we have performed some quantification methods together with the Netherlands Cancer Institute. So where we saw very few mesotrax residues in the final T cell products. That's also because during the formulation, so we have to wash the cells and all these residual drugs, and they will be mostly washed away.
Thank you. Got it. And I think another question about selection. So are there any selection or control for the heterogeneous allele editing? For example, is a gene insertion on one of the TCR alpha allele only?
Yeah, we have performed some preliminary results. So where we want to quantify how many cells they have double analytic locking, how many cells they have single analytic locking. I think according to our results, the majority of cells that only have single analytic lockings for the TCR integration into the TCR alpha locus. Of course, a lot of unlocking the neo, they are most likely to be disrupted, so not expressed.
Thank you. And we had a question about the CRISPR system. So a question is, is on how did you ensure that the CRISPR-Cas system used does not permanently stay in the target cells, and how did you inactivate the engineering tool?
So the way we engineer cells, we electroporate the nucleus proteins. So these nuclear proteins, they only have very limited time spans inside the cells. So the majority will be degraded very quick.
Thank you. Yeah, so I think it's pretty standard now to use the protein and get RNA format. Yeah, and I'm trying to select. There are a lot of questions about MTX selection. Let me try this one. So, does the MER6 epitope interfere with the TCR expression or activation?
Yes, we have compared. Yeah, so this is also our concerns at the beginning. So we have performed the head-to-head comparison between the fully human TCR with the MER6 modified TCR. And so in terms of the TCR expression and T-cell activation and T-cell functionality, so far we did not observe any adverse effects with introducing the MER6 epitope into our TCR sequences.
Thank you for the answer. And my last one is I want to add in my personal question. So, we have a lot of people talk about personalized CAR T-cell therapy and also more allogeneic therapy as well. So where do you see the future for individualized like TCR-T therapy versus allogeneic sort of therapy? Do you think maybe one day the individualized therapy can be more affordable and available? And basically, a timeline or your future on that aspect.
Yeah, I mean that's a great question, and we are always thinking about so what's the future of the kind of TCR, kind of adopted TCR kind of therapies. Of course, I mean so right now it's a very lengthy procedure. So we have to perform the screening to identify the TCRs, and we have to perform the editings. With multiple TCRs to treat a single patient, and that's for one single patient probably will we have to spend more than 3 or 4 months. I mean to perform all these procedures. So I think the field is advancing very fast. So what we are trying to do, I mean, at Neogene is to optimize every single procedure of our screening and manufacturing to make them as efficient as possible and as cheap as possible. So I think one of the approaches we are taking now, I mean, is trying to make the screening as fast as possible. And in the future, maybe there will be a screen-free kind of TCR discovery. So we don't do any kind of screening anymore, can just use some, in silico methods to get the TCR sequences. And when with next generation kind of TCR editing platforms, so we'll also be able to manufacture a lot of cells in a very short time period.
Yeah, I definitely agree. I think lowering down the cost and shortening the timeline and potentially, having more GMP materials available to make everything fast turnaround time will be essential to move the field forward. And thank you very much, Dr. Kong, for your answers. And thank you very much for the audience to join our session. And we look forward to seeing you in our next talk. Thank you.
Thanks a lot.